Hi, everybody. Dr. Phil from MedPathway. Today, we're going to look at gene expression, and then we're going to do a couple of workshop passages that are going to apply experimental data to actually understanding important topics in gene regulation. We'll have a prokaryotic example, a eukaryotic example, as well as some other stuff. So let's take a look. So this is Crick's central dogma. I'm going to argue this is where gene expression actually was born conceptually. So we know that Watson and Crick in the 1950s solved the structure of DNA. And then Crick immediately dreamt up the idea that RNA would be an intermediate between DNA and protein. That is, in order to access the genetic code and to translate it, you would need what he called messenger RNA. But so that was just a hypothesis. It wasn't until the 1960s, the early 60s, that Francois Jacob actually physically isolated messenger RNA, showing us that Crick's hypothesis was validated. But then the world was turned upside down when Baltimore and Temin isolated reverse transcriptase, an enzyme from retroviruses. And so what you see here is that they show that RNA can be converted into DNA. Kind of a heretical topic, but what we found out was that the reverse transcriptase enzyme has multiple enzymatic functions. So it can take a single-stranded RNA molecule, convert it into a DNA RNA hybrid. And what you see here then is that's a substrate. It's actually an intermediate in the viral life cycle. That substrate can then be acted upon by reverse transcriptase again, where it chews off the RNA entity, creating a single-stranded DNA substrate that's then used by RT for its third enzymatic activity, which is to make double-stranded DNA, which can then integrate into the viral genome as part of its lysogenic life cycle. So then a reverse transcriptase has multiple enzymatic activities. But in order to go from a DNA molecule into a protein, there's a lot of regulatory steps. Now, each of these steps is highly controlled, and there's a number of diseased processes that occur at each one. So the first one is transcriptional control. This is where DNA is converted into an immature RNA known as hnRNA, this is heteronuclear RNA. And so this is a RNA molecule that has not been spliced or capped or poly A added to it. And so when you do these events, you make a mature messenger RNA, and then it's ready for nuclear export, which in itself is a regulatory event. And then the messenger RNA in eukaryotes is going to be cytoplasmic, where it can serve as a substrate for ribosomal translation, or it could actually be degraded. So the stability of the messenger RNA molecule is an important feature of its regulation. But you know, these nucleosomes are going to be barriers for all the transcriptional apparatus, because we know that in eukaryotes, the organization of the genome is in this beads on a string model. And what you see here is a histone octamer core, like a spool where the DNA thread wound around it is actually stored and is compacted in various forms in order to protect the genome. So what you have to do is you have to open it, you have to close it. And so one way to access it is to have these histone tails that occupy the histone octamer. They can be methylated, they could be alkylated. There's a number of variations that can happen, like phosphorylation, acetylation. And what this serves to do is it serves to as a code for reader proteins to choose to open it up and let the transcription apparatus in, or to close it and shut it off. So if you're open, you're euchromatin. If you're closed, you're heterochromatin. So when you look at these messenger RNAs that are made in prokaryotes and eukaryotes, there's some similar yet different features. They both have +1 sites. These +1 sites define the first RNA base pair laid down by the RNA polymerase. Upstream, you see these cis-acting elements that serve as promoters. There's the Pribnow box in prokaryotes. There's the Tata box in eukaryotes. But eukaryotes also have distal elements known as enhancers. And what these things can do is lie thousands or even millions of base pairs away from the actual +1 site, and they can serve to actually generate these higher order chromatin structures where you see things like looping. And so here's an enhancer binding to its cognate transacting elements, and then it coordinates with RNA polymerase and TF2D, which is a top-top binding protein, to generate these transcription complexes, which can be active or inactive depending upon a number of things, including the tissue, cell type, point in development. So looping occurs in prokaryotes, it includes and eukaryotes. Today, we're going to see looping in the arabinose operon, which we know has been on the MCAT before. We're going to see some very interesting experimental data and have you decipher what actually the looping is doing with respect to E. coli's regulation of arabinose catabolism. So when you look at the actual enzymes, the RNA polymerases in prokaryotes and eukaryotes, what we see is that the prokaryotes, it's a much simpler, as you might imagine, version. So there's one polymerase, has 4 subunits, as you see here, but there's a sigma factor that actually can be added in as the 5th subunit to change the apo enzyme to the holoenzyme. And what you see on number 3 here is that there's various drugs that can inhibit prokaryotic polymerases, and those are great antibiotics. In contrast, eukaryotic RNA polymerases, there's 3 of them. Probably the most important one is RNA polymerase 2, because it regulates messenger RNA, unlike polymerase 1 and 3, which regulate ribosomal and tRNA respectively. And then we know that TF2D, the top-top binding protein, can recruit polymerase to its promoters, and then also RNAP2 is inhibited by alpha amanitin. So when the polymerase gains access to its promoter, what it does is it can actually help separate the 2 strands, and then it generates a new messenger RNA transcript in the 5' to 3' direction using the anti-sense strand as the template. The sense strand is identical to the newly synthesized message, with the exception of uracil for thymine, as we know. So how do you process RNA? So here what you see is a generic RNA molecule, an hnRNA that just got made off of RNA polymerase 2. So at the 5' end, we have a cap. At the 3' end, we have a poly-A tail. But what we see here in blue and red are a number of exons, and there's also introns in between. Now, the top transcript right here actually includes all the exons, including the red one. But the bottom transcript actually skips the red exon. And so any one message can potentially be alternatively spliced to make multiple different protein products. And so choosing which exon to include or which one to skip is actually a way to diversify the number of proteins derived from any single message. We can further regulate these messenger RNAs by taking a look at poly-A site addition. So in this exon over here, what we have is cis-actin elements that have cleavage spots for RNA nucleases. And so they cleave. And so you see down here that once you cleave, you direct the poly-A polymerase to put all its A's on at the end of the 3' part of the tail. So what you have in this hypothetical message down here is alternative sites of cleavage. You can cleave at the promoter proximal, they call it the PAS, or the promoter distal. When you cleave at the promoter proximal, what you do is you truncate the messenger RNA. You leave out 3' untranslated material. The actual coding sequences will not be altered in most cases. If you choose to actually cleave at the distal end, what you do is you include additional 3' regulatory elements that actually serve in many cases to bind microRNAs. And these microRNAs impose another level of translational regulation onto the message. So it turns out where you cleave matters. Cancer cells are cleaving at different spots than normal cells. And we've made some passages up that show you that actually where you cleave and how you process matters under a normal as well as a number of pathological conditions like muscular dystrophy and mantle cell lymphoma. So here what we get then are 2 different types of transcripts in eukaryotes and prokaryotes. We have in eukaryotes, we have the 5' and 3' untranslated regions, the 3' A tail, the 5' cap. This cap serves to actually recruit ribosomes, and then the ribosomes do Kozak scanning where they diffuse in 2 dimensions until they find the first AUG that's in frame, and they insert methionine in there. In contrast, prokaryotes don't have the cap, do not have the 3' poly A tail, but what they do have are some sequences upstream of the initiating codon called Schindel-Garno, GGAGG, that's actually complementary to the 16S ribosomal RNA of the ribosome. And so that's how you recruit the ribosome. And so what you see here then is on the right, what you see is that you recruit through the Schindel-Garno, the 30S small subunit, and then the big subunit comes in. Concomitantly, you hydrolyze GTP, you recruit the initiating methionine, and what you end up doing is you make your loaded ribosomal 70S complex onto the actual message itself. But what you should recognize in prokaryotes, they're polycistronic. That is, the RNA polymerase is transcribing while the ribosomes are actually translating. And you can do this because both events occur in the actual cytoplasm. But in contrast, eukaryotes have spatiotemporal regulation. They actually make the message, splice, and process it in the nucleus, and then they export it out into the cytoplasm, where then you have, in a manner analogous to prokaryotes, you have the small subunit coming on before the big subunit, and then you recruit the actual tRNA methionine initiation codon. So we're going to look at the first model of gene regulation. This is the classic lac operon or lactose metabolism. It's specifically listed in the double AMC content outline. It's called the Jacob-Monod model. And so what we see then is that Monod learned in the 1950s that when he grew E. coli on glucose and lactose, it had dioxic growth. That is biphasic growth pattern. Over here, you see A595 represents growth. So what would happen is that the cells would actually use glucose first, and then it would, after glucose was depleted, it would use lactose. And what he figured out was that genetically he figured out was that the lactose operon, what it does is it only uses lactose, well, when lactose is available, with the exception of glucose. So glucose goes first. And so you have three genes, Z, Y, and A, where Z is beta galactosidase gene, and it actually cleaves lactose into galactose and glucose. So in the dioxic growth pattern, they figured out in that phase 1, there's very little lac-Z. But in phase 2, when the glucose is all gone, then lac-Z is heavily expressed. And what they figured out through genetics was that in the presence of lactose but no glucose, that lactose binds the repressor. And when the repressor is bound to lactose, it can no longer bind to the operator sequence in the operon. And as a consequence, the RNA polymerase was able to bind. But if the polymerase bound, the repressor couldn't bound. So they're binding at the promoter and operator region at the 5' end of the operon is mutually exclusive. So in the presence of lactose, what you have with no glucose, what you have is Z, Y, and A can actually be transcribed. I'm not going to mention what happens in the presence of glucose and galactose with respect to what's known as catabolite repression, but that's something you should probably be aware of. So what we did was we actually designed our lactose operon passage on the biotest that we have to all be genetically a genetic focus, because that's what Jacob-Monod did with respect to the lactose operon. There's also the tryptophan operon, and this uses a slightly different variation known as the attenuation leader sequence model. So what happens in this case is that in the 5' end of the operon, what happens is that you have this leader sequence here. And so the idea is that under low tryptophan conditions, what you'd want to do is you'd want the E. coli cell to produce the genes that actually make tryptophan from scratch. But under high tryptophan concentrations, you don't need to be making tryptophan. So what would happen then is by having this leader sequence endowed with a number of tryptophan codons, and in fact, tryptophan is kind of a rare amino acid. So having several tryptophan codons in the leader sequence is kind of an interesting regulatory feature. So what would happen under low tryptophan conditions, you'd expect there to be low tRNAs loaded with tryptophan. And so what happens is the ribosome stalls and is sitting there looking for tryptophan tRNAs that are loaded up. They can't find them though because it's low tryptophan. And so what happens is that you generate this hairpin loop here from the red region and the blue region. These are complementary sequences. And what that does, it allows for the RNA polymerase to actually produce transcripts. But on the other hand, during high tryptophan, the tRNA tryptophan codons, they're not low at all. So as a consequence, the ribosome can move through. And when it moves through, this region 2, region 3 loop can no longer form, but a separate region 3, region 4 hairpin loop forms, and that terminates transcription. So the position of the hairpin loop as a function of tryptophan concentration, which is sensed by the tRNA tryptophans, is what determines how tryptophan attenuation and leader sequences work. And we have a good passage on this as well. So tRNA charging. So what we have then is an amino acid and an amino acyl tRNA synthetase. This enzyme is remarkable because it can recognize a certain amino acid and it can actually modify that such that it hooks up to the precise transfer RNA that's designed by the genetic code to recognize it. It requires a lot of energy. So you burn ATP to make the enzyme amino acyl AMP complex. The tRNA gets loaded on, kicking out the enzyme. And what you do is you generate this ester linkage between the amino acid and the actual tRNA. And we know that in protein synthesis, methionine is the first tRNA that's used in the vast majority of cases. So then how ribosomal translation works is that we load the methionine tRNA into the actual ribosome at the P site. So there's the A, the P, and the E. A is amino acyl, P is peptidyl, and E is exit. And then what happens is second one comes in, and what you see is good base pairing here between the codon and the anticodon, and you juxtapose these two amino acids together. And as what happens then is you actually translocate and you actually make the peptide bond, opening up the A site, and then the new one can come in here. This is actually inhibited in eukaryotes by cyclohexamide. We have a physical sciences passage on ribosomal footprinting, this really cool technique where you can actually isolate frozen ribosomes on messenger RNAs, and you can get a lot of information about which codons are used, especially this one called selenocysteine, which is the 21st amino acid. So here's the peptidyl transferase organic reaction here. What you see is you juxtapose phenylalanine and methionine here, and you have a nucleophilic attack by the lone pair on the nitrogen into the electrophilic carbonyl. You make a tetrahedral intermediate, and once it collapses, you're basically making an amide, and you're releasing the alcohol is what you're doing. So you're basically taking you're making an amine into an amide. You're getting rid of the ester bond, and you make it right here's your amide, here's your peptide bond. Okay. So let's take a look at some gene expression. Now, we mentioned northern blot before, and we talked about that northern blot was to RNA what southern blot is to DNA. And so here's what you can do. You can run out in a bunch of lanes messenger RNA samples from a variety of sources. It could be a patient, a control, it could be a number of things. And what you have to do is know that there's a DNA sequence that you want to probe with. And so by radiolabeling the DNA, you see with the asterisk here, you can probe these single-stranded RNAs that have been fixed in situ on a membrane, and you can generate autoradiographs that are going to light up the regions where there's hybridization. But northern blots are cumbersome, and what people have done is they've largely superseded them with PCR techniques. And I think PCR is an incredibly important technique. So let's take a look at PCR techniques. So here is PCR, and I'm going to argue that traditional PCR is not very quantitative. So we're going to transfer over to real-time PCR in a second. So what you have then is a duplex DNA molecule, okay? This duplex DNA molecule can be heated. And when you heat it, you dissociate the strands, and then what you can do is cool the strands in the presence of primers such that they anneal to their complementary sequences. You remember but all DNA polymerases use primers. In vivo, they're RNA primers. In vitro, they can be DNA primers. So one of the things to consider when designing a PCR experiment is how to make your primers. Well, first of all, they have to be complementary. They have to be a sufficient length and a sufficient content of nucleotides such that they can bind, and they don't bind to themselves. They don't form hairpin loops that prevent themselves from binding to the actual parental DNA strands. So once you've achieved that, you can extend with DNA polymerase, and what you do then is you amplify from one DNA molecule to two DNA molecules, as you see here. But it's not quantitative. Generic PCR, you can exhaust your reagents by hydrolysis, and so as a consequence, you're not going to get an actual quantitative event. But RT, or real-time PCR, or quantitative or qPCR, is actually quantitative. Let's imagine that you wanted to measure the amount of transcript in a certain cell. So what you would do is you would isolate the messenger RNA, the poly-A RNA, okay? That would get rid of your transfer RNAs, your ribosomal RNAs. And then what you do is you'd use reverse transcriptase to make a complementary DNA template. And then what that would serve is a double-stranded DNA substrate to perform real-time PCR. So what you've done is you've taken your messages and you've converted them into double-stranded DNAs. And so the more messenger RNA you started with in the cell, the more abundant its expression, the more cDNA templates you would have as substrates. So here what you do is you perform PCR. I'm only showing you the top strand, and you perform it in such a way that you have primer that recognizes your gene of interest, but you've also placed this interesting probe inside the gene itself, in the body of the gene. The probe has been tricked out such that at the 5' end, you have a floor, and at the 3' end, you have another floor, such that you shine light and it hits the first floor at a certain wavelength, it fluoresces, but the second floor can quench that fluorescence and you would never detect it. But what happens is that as the Pac-Man polymerase comes charging through right here, what you know is that it has a 5' to 3' polymerase activity, but it also has an exonuclease activity, and it can chop up this internal, these internal probes. And when it does so, it clips it such that you release the 5' floor, and as you shine light on it, this one has been dissociated far enough in space that it can no longer quench it. And then what you can do is you can start measuring fluorescence, and then at a certain number of PCR cycles, you can detect an actual threshold number of fluorescent events. And so the threshold is considered the minimum that it takes in order to detect your signal here. And so what happens then is that the more PCR cycles that you go to, the less input DNA you had in the beginning. And what you see here is a green one here that has threshold at an earlier cycle number. That means there was more DNA to start with. So the PCR cycle that you achieve threshold with is inversely related to the initial amount of substrate template you had in your PCR reaction, which means you had less messenger RNA in that cell. And what you can do is you can quantitate by the difference in threshold number, because you know that in each PCR cycle, you go up by a factor of 2. So with that in mind, we're going to start some workshop passages. We have some really challenging stuff today that's going to make you take all this basic information, apply it to an experimental context. We want you to learn how to interpret data, to read data, and to actually come to important conclusions, because that's exactly what the MCAT is doing. It's asking you to interpret data